Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm very excited to be here today again with Dr. Slavé Petrovski, who's Vice President of AstraZeneca's Center for Genomics Research. If you saw us live at ASHG or if you listened to the live recording that we put out a couple of weeks ago, then you'll know Slavé very well. If you haven't, I'd encourage you to go back and listen to that before or after this episode. We're going to give context on what Slavé does and some of the amazing work they do at AstraZeneca, but I highly encourage you to listen to that. So at this point, Slavé is a longtime friend of the pod as a second episode. We're going to dive in detail into some of the amazing work you've done, big population genomics asset and software called MILTON, a lot of the work you've done to drive actually sustainability and energy savings through the large-scale pipelines that you run and other insights that you've had from large-scale diverse population genomics programs like UK Biobank, FinGen, and also other programs that we'll talk about today. So Slavé, thank you for joining us.
Yeah, no, it's a real pleasure to be here with you, Patrick. I've been following you and this podcast for some time, and as I've mentioned before, it's one of my favorite channels to keep up to date with the beautiful world of genetics.
We will keep them coming. I want to start actually with your origin story that a lot of people probably wouldn't have heard. We'll talk a little bit about your work today, but how did you actually get into science and genetics in the first place? I think you had a pretty influential conversation with your dad that I'd love for you to tell people about if they haven't heard the story before.
Yeah, no, not many people have heard this story, and yeah, it is quite interesting. I like the term the origin story. My career took a very interesting turn early on. I was originally enrolled in a Bachelor of Business Information Systems. And I was about a week into that course, and I had a casual chat with my father, and he gave me a simple but what turned out to be kind of a career-defining piece of advice. And all he said was, as long as you are enjoying what you're doing, you'll be successful in whatever job you do. And I kind of interpreted that as a message, follow your passion, right? So that next week, I made the bold switch. I went from what I thought would be kind of a safe career, business information systems, very promising opportunities in terms of jobs, to a double degree in Bachelor of Science, major in genetics, and combined it with a Bachelor of Information Systems. And bringing these two worlds together, back then, young me felt like a risk, but it was just, looking back, it turned out to be, I would say, one of the best decisions I've made. I was able to get that unique blend of genomics, biology, while also tapping into advanced computing systems and getting formal statistical learning training. And it's kind of shaped every step of my career since then, really.
That's an amazing story. We actually have that in common in a very similar way. I started studying math and economics, got a little bit more interested in biology and disinterested in economics. I had some very similar advice from my dad at the time that if you do math, you can do almost anything after that. If you're liking it, go for it. And then I followed the biology trail as well. And I think we have to give a shout out to dads or moms who give that kind of career advice early, because it's really helpful, other than, go be a lawyer or doctor and make a lot of money, which unfortunately is still the advice often these days.
And also, to your point, we also have to be mindful with our own children, right? These little small comments can have such profound impacts on their life choices. And so it really is important that we spend the time with our children as well in advising them properly.
Yeah, I couldn't agree more. So you followed that thread all the way through to where you are today. Maybe you could talk about the decision to go from academia to industry, because a lot of people listening to this podcast have made that transition already, or might be thinking about it, or maybe thinking about going back. And maybe you can talk a little bit about what drove you to make that decision at the time, and how you think about it today as well.
Yeah, unsurprisingly, that was another key turning point. I mentioned the conversation with my dad, but then this decision was similarly profound in terms of where my career went. And again, I can't take all the credit because honestly, I spent quite a good time speaking with many mentors before finally making that decision to join AstraZeneca. I had a comfortable academic faculty position at the University of Melbourne, working with some of the brightest minds in genetics and clinical disease, especially around epilepsy genetics. And I enjoyed all those freedoms to explore ideas, mentor students, write grants, and contribute to knowledge creation. I, probably like many of us who've made the transition, owe a lot to academia. Those experiences really teach us to ask deep questions, to think critically, and very importantly, focus us to build evidence-based narratives. These are skills that have remained just as critical now that I'm in industry. But I took the role at AstraZeneca right around the time that AstraZeneca launched its company-wide genomics initiative. This was an initiative motivated by the belief that there is no better model to define our therapeutic strategies on than humans. And so AstraZeneca's vision was to build one of the most comprehensive human genomic and multi-omic resources to really help us answer some of the most important questions across the value chain. And importantly for me, in a very patient-centric way, it kind of just felt like a perfect fit. Doing what I love to do within academia, but now bringing it to industry, scaling that up, and being closer to patient impact. Now, naively, I thought when I made that transition, it would be great exposure for a few years, and then I'd learn and be able to return back to academia. I think it's safe to say I haven't yet felt like I'm done learning quite yet. So feel like I've still got a lot to learn. Also, another important point, I think you probably reflect on this as well, Patrick. The lines between academic and industry research are becoming increasingly blurred. And industry for a long time has absolutely benefited from academic research. And today it's just taking a growing role in helping shape that research too. Academia is critical for pushing boundaries, especially in critical foundational studies. Keep chipping away at the vast unknown. And then industry looks to really translate those discoveries into patient impact, often at a scale that's just difficult to achieve elsewhere. So, it felt like a really great transition for myself. And I've been in AstraZeneca now for about 8 years now. Moved over to the UK in 2018. And the role itself that I have in AstraZeneca has evolved quite a bit. It's been quite a learning experience.
Yeah, I can imagine. What you've worked, I think from the early days, you've had access to some of the biggest biobanks, resources. You've funded the creation of a lot of these. You've also, I think, pushed the envelope in sequencing and helping to sequence populations that are underrepresented in research. I'd love if you could just talk through some of the major things that you and your team have learned from both the UK Biobank, FinGen, some of the known names, but also maybe some of the more niche programs you've helped fund and get off the ground, and how that relates to the drug discovery work that you focus on every day.
Yeah, for me, one of the most important learnings working with these large biobanks that are really blossoming in the last decade, I think it's all there to see in Nature, Science, Cell, and every other journal, is the absolute importance of scale to get high quality, robust discoveries. And for us, within AstraZeneca, especially with a strategy that is focused a bit more on large effect variants, so it tends to be much rarer genetic variants, and what impact they have on human health. These variants tend to be individually rare, so it's hard to get statistical confidence from individual variants, but collectively they become quite powerful when aggregated in meaningful ways. Like, for example, what are the effects of all loss of function variants in gene X on human health? And you look across the population and find all those individuals that have a loss of function variant, and then correlate that with their health outcomes. And to be able to draw robust conclusions from these kinds of studies, what's been critical is kind of the scale, scale. The other aspect is diversity, as you mentioned. In 2021, about 3 or 4 years into my tenure at AstraZeneca, we looked at the composition of the Genomics Initiative database that was fueling our internal R&D. And at the time, about 92% of the sequences came from individuals of European genetic ancestry. Now, at the time, this was about 3% higher than what was cited as the global average, but I think we recognized it was still far from good enough, because we knew, like many others, that to really truly understand human biology, we need to be studying all humans. And so, we had the interactions with our senior leadership and got the support to be able to identify and establish partnerships with historically understudied and therefore, by extension, often underserved communities across the globe. And now we have, strong partnerships with Mexico City, in South Asia, in East Asia, in Africa, and various other parts of the globe. And for us, this wasn't just about the obvious ethical benefits. As a result, we are today already tapping into new discoveries in these diverse populations that simply would not have been possible if we continued to focus on one global community. You're finding rare genetic variants that are maybe not so rare in particular parts of the globe because of adaptation, because of various factors. And you can kind of escalate the understanding of human genetics by expanding beyond the one well-studied community. And I really, from our experience, can't understate the importance that diversifying the ancestral composition of our database has had. We're leveraging now over 1.7 million research participants in our research within AstraZeneca. These are individuals where we have sequence data linked to rich medical histories, mostly longitudinal. And today, about 40% of the research participants represent individuals of non-European genetic ancestry as a result of these intentional efforts to build partnership, identify, build, and maintain partnerships across the globe. And firsthand, this has been quite invaluable in terms of helping us discover new drug targets, for tomorrow's medicines, helping us better understand disease progression across global populations, helping us discover new biomarkers for our medicines, helping us understand the heterogeneity, the kind of differences in response in our patients to our medicines. And also, we'll touch a bit about this, I think, later on, because it's some recent work that we've been doing, helping us identify the right patients for our medicines much earlier on to really ensure that these individuals benefit from the medicines earlier on in their disease, rather than kind of closer to end stage disease.
Yeah, let's talk about that right now, because it's one of the directions I wanted to go next. How are you using these biobanks and the internal data you have to answer those kinds of questions?
Yeah, so with the UK biobank in particular, I think that was being quite fundamental, and it is in a lot of ways the top shelf example, because it's got the scale, and it's got the clinical breadth. Now, it is lacking a little bit in diversity, obviously. I think 92 to 95% European genetic ancestry. But to be fair, 5 to 7% of non-European, of 500,000 is quite large number. You get into tens of thousands of other ancestries. But it is a dimension where I think the other biobanks have been quite helpful. Yeah. And so being able to mine that richness of genetic data from 500,000 plus individuals, linked to health outcomes, linked to biomarkers, has really allowed us to identify new candidates for drug targets. There's over 16 programs that have been launched as a result of human genetic discoveries across the therapy areas that are progressing through. It's really helped us identify new clinical biomarkers for some of our existing programs. And to an extent also, it allows us to better understand earlier what are some of the potential on-target safety liabilities. When a lot of these medicines look to, for example, inhibit a particular protein, and when you've got access to millions of individuals, you will find individuals in the population that have these natural, rare genetic perturbations that in a way mimic what the therapies are trying to do. And you can try and see, are there potential clinical signals in the population that could be important in how we progress these through the pipeline. I think the one that's quite exciting is most recently is the work that we've been doing integrating the genomics, proteomics, and biomarker data from hundreds of thousands of individuals in the UK Biobank to develop AI models that can successfully predict the onset of over 1,000 disease endpoints, sometimes up to 15 years before they were clinically diagnosed in those individuals. And for me, this kind of opens up the critical transition opportunity from the largely current state of affair of dealing with disease to being more proactive and intervening, detecting disease at its earlier stages, intervening earlier to really maximize healthcare benefits for society.
If you look across the broad set of biobanks that you work with, are there any hidden gems that people aren't as familiar with that you think either are really impactful today or have the opportunity to have a really big impact in the next few years that maybe aren't as on the radar as UK Biobank, FinGen, or some of the others that we talk about regularly?
Well, to be honest, I would say all of them.
Yeah.
And I know it's an easy response, but population genetics as a discipline, I'm sure you've had your own training in that field, has been in my mind underappreciated in terms of the impact it can have on understanding clinical health. And one of the things that really opened my eyes up with starting with the UK Biobank, but also FinGen, Mexico City, and others, is that by studying a population, rather than historically what we do is we are interested in kidney disease and we would ascertain people with kidney disease, typically tertiary care settings, near end stage 5, dialysis, and then we'd look at genetics to understand why they got sick. One of the real powers of population genetics, and where I think my own kind of career got very, very interesting in the last half decade or so, is that because we've now got these population ascertained sequences from these collections of global biobanks alongside the compute power available today, we can actually start to ask new questions. For example, what are the genetics that preserve organ health? What are the genes that slow down disease progression? What are the genetics in the population that make certain individuals resilient to certain disease biology? In a way, this kind of flips the traditional discovery paradigm on its head. Rather than using genetics just as a tool to help us understand why people get sick, we can now use it to also learn from those lucky individuals who've won the genetic lottery for weight management, respiratory health, and so on. Think about those friends you have who somehow remain lean despite living in an obesogenic environment, who have been heavy smokers for many, many years, yet somehow they manage to maintain healthy lung function. PCSK9 is perhaps the most well-cited example of this, the kind of concept of genetic resilience, whereby individuals in the population with genetic loss of function variants in PCSK9 are observed to have significantly lower cholesterol levels and also significantly reduced cardiovascular risk compared to their population peers. Now, this discovery in the kind of human population catalyzed years of preclinical research, and today there are many companies exploring therapeutic PCSK9 inhibition in clinic to help benefit all those individuals who weren't quite as lucky as those natural PCSK9 carriers. And so today, with these biobanks like UK Biobank, Mexico City, FinGen, all of us and many others, we're able to actually do this more systematically. And it turns out there are many, many more examples like PCSK9 in nature's kind of treasure trove of natural clinical trials. In the UK Biobank in Mexico City, for example, we discovered, combining those two biobanks, individuals with rare genetic perturbations in MAP3K15 have a significantly reduced risk of being clinically diagnosed with diabetes in their lifetime. If we look at the population data, we also see that individuals with rare genetic perturbations in TSLP have significantly attenuated eosinophil levels, which is kind of consistent with what we know now from therapeutic TSLP inhibition. Individuals with IL-23R genetic perturbations, that they appear to be shielded from multiple autoimmune conditions in their lifetime. And this list goes on. It's an area I'm really passionate about, so I can kind of talk about this all day, to be honest.
Yeah, I actually do want to follow up on that because hearing you talk about that, these seem like they open up a much broader aperture for the chemists and the drug discovery and therapeutics development people you work with, right? Versus a target that we know if you have a loss of function, it causes disease. I've got to imagine that you just open up the creativity of how do you go after this pathway or use the biology to just think a little bit more creatively versus directly at the target. Is that right? And if so, how do you collaborate with the rest of the organization to do that?
Yeah, it is kind of right. I think, when we find genes that cause disease, they are critical, right? And I can't underestimate that because it gives us new insights into biological processes and pathways. It kind of helps us understand what are what's the pathways involved in disease, and then how can we intervene. Now, when we identify rare genetic perturbations that actually kind of protect or make people resilient to certain disease biology, in a way, we're shortcutting things. We're no longer needing to try and develop medicines that reverse the cause of disease. We're now saying, okay, there's people in the population who are naturally resilient to immune diseases, to metabolic diseases. Let's find the genetics that's making them resilient, and let's try and mimic it therapeutically. Let's try to develop medicines that mimic that. PCSK9 was one of those great examples. But what's striking, though, is that many of today's medicines, when they were being developed, at the time, we had no clue that they were supported by this kind of genetic resilience evidence that we see in the population. But now with these big biobanks, we live in a very different world. Just in the last 5 or so years, to be honest, with the scale of the biobanks and the sequencing resolution of data that we have available today, rather than some of the more traditional technologies, it's clear that for many of the medicines that are actually launched and helping patients today, nature's very own clinical trials already validated them long before they reach clinic.
Yeah.
I mean, if you go to our azvwas.com portal and you look up TSLP, the gene TSLP under gene level analysis, again, this is a statistical test that aggregates the effects of many rare genetic perturbations seen in the population in that particular gene. And then if you just click, we have a radio button that says odds ratio less than 1. It allows you to then all of a sudden focus in on the kind of clinical phenotypic indications that are significantly lower in individuals with natural genetic TSLP perturbations. Then if you browse that kind of, if you like, the cream that rises to the top, so the phenotypes that are among the top dozen or so ranked phenotypes for it, you see actually does quite a good job at recapitulating what we now know from therapeutic TSLP inhibitors. For example, individuals with rare genetic perturbations in TSLP are around 42% less likely to be diagnosed with asthma in their lifetime compared to their population peers. And this is evaluating something like 20,000 different disease endpoints, and it's picking up at the top-ranked protective signals, things that we now know from clinical evidence. And we can switch it to continuous traits, biomarkers. And again, looking at it, it recapitulates with quite striking specificity what we now know from clinical evidence, that those individuals with natural TSLP genetic perturbations also have significantly lower eosinophil levels and percentages than their population peers. These are obvious today based on all the data we have from clinical programs across industry. But imagine if we had access to those data 10, 20 years ago. It could have helped accelerate a lot of these medicines to patients. So there are many more examples like this in cholesterol, in other areas as well. And I think it's just been such an exciting opportunity to look at population genetics as a tool to help us, in a way, identify new targets that confer benefits to human health, particularly for certain indications of interest where there's large unmet clinical need, and then think about how could we develop medicines in the lab that mimic what some lucky people in the population have inherited.
Can you talk a little bit about MILTON, the tool you developed? We discussed this earlier in brief, but biology is such a complex system that sometimes we go big and scaled and we find a single individual or group of individuals that have this incredible phenotype. But more often you're having to look at a lot of disparate interacting signals, and you've done an amazing job building a machine learning framework for looking at disease prediction, integrating a lot of different signals. I'd love if you could talk a little bit about that, how you developed it and how you use it today.
Yeah, absolutely. I think MILTON, to your point, is a great example of what is possible when you really start integrating the multiple modes of biology and omics that we can generate alongside the longitudinal health records that we have on these individuals. MILTON was built on the UK biobank dataset, largely because it was the largest biobank dataset that we had at the time, but also it was increasingly growingly rich in biomarker data, routine biomarker, and then proteomics and so on, and imaging data, and it goes on. And for me, when we were developing MILTON, I should actually give you the background story because it is an interesting one in itself. The original intention of MILTON was we recognized that in these big biobanks, the phenotyping is good, but there will be some mislabeling, and there will be some individuals who are cases but are missed for whatever reasons, and so we refer to them kind of like cryptic cases. They have disease, but they're labeled as not having disease, and we wanted to see if we could improve our genomic discovery yield by identifying signatures of disease in the population of patients who are labeled as having disease, and then looking in the rest of the population, in the UK biobank population, and seeing if there are other individuals that have that same signature, and whether if we augment the statistical test by removing them from controls and moving them to cases, do we, for example, for well-established positive controls, get improvements in power and significance, but also, does the effect size remain robust? And it turned out it worked remarkably well for many diseases. But as we were exploring the results, actually, we noticed what we developed was also a tool that was able to predict disease, not just current disease, but due to the nature of UK Biobank, we had 15 years of healthcare follow-up after the specimens were collected that we're doing the molecular profiling on. We were also able to predict future disease, future disease risk. And so it kind of took a whole new direction for MILTON, whereby now when we look at it, we can actually use MILTON to identify signatures in the biology, a combination of genetic, proteomic, routine biomarker data that are happening in individuals many years before they're symptomatic. Wow. And this has obvious implications for what's going to be really powerful in shifting the model from reactive sick care, where we're looking to really, as I mentioned before, manage symptoms of disease, to one that in the future focuses more on proactive healthcare, prolonging the healthy life of our society. And if we can detect these diseases earlier, ideally, pre-symptomatically, and we can intervene earlier, whether that's lifestyle monitoring, targeted therapies, it means that we'll be able to, for many of these diseases, get better outcomes, reduce the burden on healthcare systems by not relying on delayed diagnoses. And so this is a real key part of the future vision for us, in kind of shifting the focus from managing sick care to more kind of pre-screening healthcare focus, not waiting for disease to show up, but really kind of intervening as early as possible with all the tools we have available to us as a society, to really minimize the burden on kind of late stage healthcare systems, hospital settings, and see how much we can bring forward into, again, just focusing on that healthcare, the longer, healthier lives for society.
That probably completely reshapes how you think about clinical trials as well in some of these populations, right? Maybe you can talk about what would change in a world where you may be able to detect disease 15 years early. It doesn't necessarily mean that is the right time to intervene therapeutically, but how do you think about that? Because it's a hard problem.
It is a hard problem, and I think it's one that we're still working towards, because it is difficult to define. It is more challenging to define clinical trials in a proactive setting, in a pre-symptomatic setting, because it takes longer, and it's difficult to be able to. We can use proxies like biomarkers, which are often very successful in certain indications. But I think equally there are certain diseases where today we're already diagnosing patients far too late. I think amyloidosis might be one good example of this, where by the time a lot of people achieve a clinical diagnosis of amyloidosis, it's difficult because it's often quite late into the disease process. If we can identify in the population individuals who are at high risk of amyloidosis based on some pre-screening tool like MILTON, right? And we can then do kind of maybe more detailed surveillance on those individuals to diagnose them and give them that diagnosis much earlier. There are today many medicines across industry that can help those patients and help them live a longer, healthier life. Again, it's that kind of healthcare component. So I think we're starting to see the opportunity for these now. For certain diseases, indications, it might be sooner rather than later, and for some, it's going to take some time. And there are some indications where today we really don't have good interventional strategies, and so then that brings its own kind of challenge of what can we do with that information. So I think there's multiple layers here. If we have therapies that can help certain indications, then tools like MILTON will be quite precious in being able to identify these patients much earlier on in the disease process, because we've already got interventions that we could look at exploring in the clinical setting to delay disease, to stop disease, maybe even reverse disease in some settings. I think the more we shift from treating people who are closer to end stage disease to earlier on in their disease biology, I have to believe for a lot of these diseases, the better the outcomes will be for them individually, and then collectively, obviously, for society as well.
Yeah. I want to talk about how much work you all have been doing on green algorithms, and more broadly, AstraZeneca has an incredible commitment to sustainability. I don't think there's a pharma company that comes close in terms of actually really living that that pillar. And I know it goes all the way to the top from Pascal's vision. Maybe you could talk a little bit about the environmental impact of the large scale genetic studies that we do, but more importantly, some of the work that you all have done to make them incredibly performant at a very low environmental footprint. Because I think it's not something we've never discussed on this program before, but I think it's really cool the way you've been dedicating time and resources to it.
Well, and I hope it is something that will get elevated interest, because I think it is critical, and it kind of ties in with everything we've been talking about with these big biobanks. We live in a different world today compared to even just 5 years ago in terms of the scale of data that we have access to. We're now working with millions of genomes, petabytes of data, each linked to rich clinical, multimodal molecular data. UK Biobank Proteomics Consortium, the pilot consortium was 50,000, soon it'll be 600,000 specimens, 500,000 plus 100,000 repeat imaging. Wow. It's extraordinary opportunities in terms of really diving deep and properly into human biology, but also, we have to face it, it's becoming a massive computational challenge. And, I like to discuss this, I like to kind of put the scale into perspective. Every phenome-wide association study can now, with sequence data, involve trillions of statistical tests. I was chatting with one of my colleagues just yesterday, and she mentioned if you try counting to 1 trillion out loud, it will take over 31,000 years now. Just to say, I haven't tried it myself to confirm whether that's true or not, so don't quote me on it.
Sounds about right.
Maybe when I retire, I'll start counting and see how far I get. But that's the scale of compute many genomics researchers across the globe are heading towards when analyzing exome or even genome-based VWAS in these growing biobanks. At the same time, getting to the heart of it, the algorithms we developed in the last decade or so haven't kept pace. These algorithms and tools were designed for kind of a different world where we had data sets multiple orders of magnitude smaller. Let's say hypothetically that to run a single statistical test on your cohort, it costs you $0.00001, right? One test costs you 100,000th of a dollar. Now, you say you want to run GWAS and you have to perform 1 million tests. That would cost you around $10. And what feels like negligible carbon emissions. Most of us wouldn't blink at that cost. Now, say we need to scale that up from 1 million tests to 10 trillion tests to study all the genetic variation we see in a cohort of genome sequences from 500,000 individuals, while also at the same time studying tens of thousands of phenotypes in that cohort, right?
You got a big matrix there, yeah.
That same algorithmic compute, if we continue to do nothing, today would cost close to $100 million.
Wow.
And I won't even cite how much carbon emission, right? So that's clearly not sustainable on many fronts. So, going back to the work that we've been doing, when I joined AstraZeneca, and I saw that the bold ambition was to analyze upwards of 2 million research participants by 2026. And, we're at 1.7, we're getting to that. We saw this as being probably one of the key challenges and one that was coming fast at us. And for us, the costs we were wanting to get ahead of were not so much the data generation costs, but more so these perpetual data storage, processing, and ongoing analysis costs, right? Analyses aren't once. You get new data sets, you get new algorithms, new tools, you want to reanalyze the data. So it's, you want to keep innovating. So we kind of set a little tiger team, if you like, to focus on how can we scale our science without scaling the emissions and the costs. Yeah. And so one of the things that we sought to optimize was effectively re-engineering the core statistical methods we've been using. And we started this by lifting the hood on multiple mainstay algorithms with the intent of effectively redesigning those engines from the ground up, stripping away all the inefficiencies that may have served the purpose in the past. A history of small, unstable data sets. We've got to kind of create all these extra checks in place. Right. And we live in a different world now with these big, high-resolution data sets. And then optimize the engines for those tasks, the stats we run most. And as a result, we just presented at ASHG, and we had some external comms about this as well, which hopefully you saw. For those tests that account for about 80% of our analyses, we've been able to reduce the compute time by over 450-fold and cut CO2 emissions by more than 99% compared to what's considered today's industry standard statistical tests, all while maintaining the integrity of the test statistic with correlations averaging above 0.99. So pretty much near perfect test stat correlations, but 99% reduction in CO2 emissions. And this is particularly important because as a field of methods development, which is one that I and many other listeners to your podcast are involved in, sustainability has often lagged in focus. Yeah. This demonstrates that our scientific ambition can coexist with environmental responsibility, consciousness, and so on, and honestly needs to, so that we can advance together. And to your point, I'm hoping some of this work that we've been communicating recently will reinvigorate a kind of new dimension of focus in methods development that sets increasing standards on responsible genomics. Because these biomedical data sets are only going to continue to grow individually, but also collectively. All these biobanks across the globe, we're going to get increasing resolution, genome sequence data, many modes of omics, imaging data, phenotype data. This problem is only going to grow. I say problem, this opportunity.
Yes, that's right. It's the other side of the coin. It sounds like maybe to just think about the 80-20 on this. Is it fair to say that within the 80, you've got the optimizations you talked about where you really don't change the algorithm fundamentally, but there's just a whole set of redundant things that we keep doing over and over that we don't need to do anymore. Another bucket of that is actually changing the underlying algorithm itself. And then maybe there's a third one, which is hardware optimizations. You could think about FPGAs or something like that. My assumption is you probably haven't touched the third one yet, and most of the optimization is just in 1 and 2. Is that right?
That's right, which puts it, makes it a bit of a scary concept in terms of how much further can we push it if we also start optimizing on the hardware. But no, you're right. It's really, again, a lot of these statistical tests have served their purpose, a really important purpose. And the scale we were running analyses on in the past, it was fine, right? It was very negligible residual cost. Nobody really blinked. But we've kind of added all this extra stuff to these algorithms and these tools, etc. And some of it's important, but not all of it.
Yeah.
And so that's what we're looking at, going back and saying, okay, well, for our workhorse, for 80% of the tests that we need to run, what can we take out and still get that 99% plus test statistic correlation with the existing industry standards? And that's effectively what we've done. And it's quite remarkable how much is in there that probably you can take out in terms of assumptions, libraries, and so forth.
You have you made a lot of your work open source. So MILTON that we talked about, the PheWAS, AstraZeneca PheWAS portal. My understanding is that these are probably used by tens of thousands of people. Maybe you can talk a little bit about how your philosophy of open science and the decisions to make so many of these tools open source and how you think about that going forward.
Yeah, and again, kind of ties back into earlier, as I mentioned, within the industry setting, we've benefited for a long time from academic research, and we continue to benefit from it. And so for us, when we do these analyses, like in the UK Biobank, publishing AZ PheWAS, publishing MILTON, we recognize that there is so much discovery in these data sets, in these results data sets, this insights data sets, and it's impossible for any one group, be it academic industry, any one institution, to really maximize the value of all those discoveries in there. So our strategy has been, to your point, we make the test statistics publicly available to really help catalyze research everywhere. And we're seeing tens of thousands of users per month accessing some of our public-facing portals. And for me, the most exciting part is not just the volume of users. When you look at the kind of maps of where they're coming from, it's in parts of the world I never would have imagined our science touching. And so this is catalyzing new research, especially the translational research, which is, in my mind, the kind of the biggest bottleneck. I think getting the genetic discoveries is becoming relatively routine with the big data sets. We're improving, but turning those discoveries into translational mechanistic biology that we can then develop therapeutic strategies on, that can be arduous. And so, working with academic collaborators across the globe has been really critical to helping us advance a lot of our own research priorities and interests. So, for me, it's just really a critical way to be able to, build up partnerships across the globe. We have research groups across the globe, not just in the academic setting, the industry, biotechs, reaching out to us because of something they saw on one of our portals and wanting to partner with us. So it's great. It's kind of really advanced and accelerated a lot of the programs that we have internally too. And then the other aspect, which I'm sure you can probably register from this, is the sustainability part again.
Nobody's redoing it over and over. Yeah.
There's no need for hundreds of institutes across the globe to, they might run it slightly differently, but to be honest, there's going to be incremental changes. So having a vanilla analysis that you can just look up, and then if you've got your own method, you can go and do a deeper dive into a particular phenotype or gene, but at least you've got this kind of vanilla analysis that will give you 80+% of the answers.
Yeah. In a world of open access tools, open data, how do you avoid groupthink, herding? Everybody's looking at the same data sets and targets. What do you do practically or philosophically within your team to try to get people to think outside the box?
Yeah. I think, yeah, groupthink is a challenging one. And it's quite natural in any fast-moving field to follow the most popular approaches. The problem is you can, to your point, end up doing what everybody else is doing. Not because necessarily it's the best way to do it, but because it's familiar. And unfortunately, sometimes it's also because it's the easiest path to get it published, right? Path of least resistance to get it published. You just use the same approach everybody else is. So within our team, we've been quite intentional again about trying to build a culture where folks feel it's safe to ask, if we're starting from scratch today, would we do it this way? Or, for example, are all the assumptions we're making here to manage the quality of what were smaller scale historic studies still relevant today in these big, robust studies of 500,000 individuals? Right? Do those little nuances in the data really make a big impact on the test statistics like they used to when we had 1,000 cases and 3,000-4,000 controls? Yeah. And it's surprisingly a powerful question. And as part of it, we often kind of swim against the grain in that we try to remove as much of the unnecessary complexity in a lot of these methods. And complexity shouldn't get mixed up with sophistication. I think in my mind, if you could explain it simply, then you're probably on the right track, right? If you have to add all this complexity to get to the result that you're expecting, then I think you're over-engineering often. And so we kind of welcome this collegial debate as part of the culture where folks can say, well, do we really need to do it that way? And this isn't about being contrarian or devil's advocate, if you like. It's simply about just staying open-minded and following the data, right? Does the data suggest that we need to add extra layers of complexity? If yes, then we go ahead. If not, then why add the extra complexity?
Yeah, I like that. And you mentioned at the very beginning that learning, doing science and working in academia teaches you a lot about how to think really critically. Something that I remember getting drummed into me in my PhD from both Matt Hurls and Jeff Barrett was, if you find something interesting in the data, you need to at first just assume it's an artifact and something crazy. You need to prove it 3 or 4 different orthogonal ways. The crazier it is, the more you need to prove it in independent ways. I think that kind of professional skepticism is really important, right?
It is, and the way I say it a little bit more candidly is, you got to kick the tires, and
yes,
if you kick the tire long enough and you can't break it, then it's probably okay.
I presented enough artifacts that I was really excited about my first year of my PhD to learn that the hard way of asking if I divided by 2 and finding out I did not in my code. So yeah, we learned the hard way from other people kicking the tires. Maybe just to close out here in that spirit of thinking simply, thinking big, long term, how do you think about the next 5 or 10 years, whatever time horizon is that's most natural to you? What are the big things that you're focused on as a team or in the industry more broadly that you'd be interested in seeing?
So, firstly, what's scary is that 5-10 years will come so fast. I think it used to feel a long time ago, but the pace of science and the pace of technology development, and we haven't even touched really on kind of generative AI and so forth, predictive AI modeling. It is already a different world, let alone in 10 years' time. But one of the things that I definitely would like to see in 10 years' time is genomics being more core part of routine healthcare. And we've been talking about this for the last 10 years, to be honest. And so now with these technologies, getting the cost of sequencing down to close to $100, and we know that at least for inherited DNA, the data really only needs to be generated once. It would be great to see more genomic screening becoming just a standard part of preventative medicine, early disease detection, etc. I think next 10 years would really love to see us make, and there is some progress in the community towards this, I have to be fair, but seeing this much more routine across the globe. On top of that, also seeing a fuller picture of health, not just the static genetics, but how can we have these, if you like, avatars, individuals where We've got multiple modes of dynamic omics, so transcriptomics, proteomics, metabolomics, imaging, digital health data from wearables, etc., all integrated, to track their health of an individual over the course of their life, rather than at the moment, a lot of these tend to be isolated clinical snapshots, usually when they already become patients. But it'd be great to have it, pre-symptomatic, so you can start to model and develop even more accurate predictions of disease and patient management care, kind of individualized, personalized, customized, however we want to call it today, to an individual. The other one I think is, again, on a similar theme, how can we better capitalize on genetically stratified trials? And here I'm not referring necessarily just to polygenic risk scores, which I think is where most of the listeners would kind of think the direction is. For me, it's more about increasingly transitioning to a model where patient care management is based on individuals' underlying molecular cause of disease, their disease drivers, rather than just based on their clinical presentation, which is still how a lot of patient management is done today. Take the success from cancers as a great example of this transition, where the field is increasingly focusing on treatment based on mechanistic causes of an individual cancer, rather than based on the site of the solid tumor itself. So how can we do this more across common chronic diseases, rare diseases, and beyond? And then just lastly, with kind of MILTON in mind, care that starts earlier, right? How can we detect diseases before the symptoms appear, intervening at a stage where we can generally preserve organ health, rather than try to reverse the damage that's been done often when we treat them in the hospital tertiary setting, because that is a bigger uphill battle with well-advanced illness than if we can maybe intervene lifestyle and various other tools that are available to us much earlier. So for me, the next decade should also be about how we, one of the biggest challenges that we have is it's not so much discovering new targets. I think hopefully through this podcast and through the literature and AZVWAS, etc., and other portals that are available, discovering new targets isn't the challenge anymore. It really needs to be about turning the most meaningful discoveries into real treatments that transform care and simply just change the lives of these patients. So, I'd like to see the next 10 years really focusing on transitioning from the discovery towards actual medicines that are impacting patient care. And there are examples of this, so it's not like it's not happening, but just seeing that escalate at the same rates that we have been successfully improving genetic discoveries themselves.
Thank you for that amazing clarity of vision. I think for anyone thinking about how they want to spend their time for the next 10 years, they've quite a few chunky options in there to go after. So I just want to thank you for the time today and all the great work you've done and sharing that with us.
It's been an absolute pleasure, Patrick. Thanks.
Great. And as always, thanks everybody for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.